Hi, this is Miss Litton, and this is my wonderful zero period biology class. Say hi.
Hi.
Okay. All right. So we're looking at this picture of these puppies, and why are these puppies so different? First, find the mom and dad puppy in there somewhere. Okay. Pick your favorite puppy. Did you pick your favorite puppy?
Yeah.
Okay. Do any of the puppies look exactly like any of the parents? No, they don't. Okay. And that's because if you're looking on your notes, this would be under 5.3, reducing the chromosome number for reproduction meiosis. This is our last box, genetic recombination. Genetic recombination is variations due to the formation and union of two gametes made by what process? Meiosis. Variations due to the formation and union of two gametes made by meiosis makes sexual parents capable of adapting to changing environments. So these puppy babies look different for three big reasons. One reason happens during prophase 1 of meiosis. And what happens at prophase 1 of meiosis? Crossing over. Okay. Could Blue tell Slate what is the definition of crossing over? Go ahead. Okay. And in our notes, come back to me. In our notes, crossing over during prophase 1 is the exchange of sister chromatids by homologous pairs. Okay, so this would be an example if you're looking through the stages. Oh, this is the video I owe you, right? Yeah. If we are looking at meiosis 1, we can see evidence of crossing over in each one of these chromosomes. That is increasing variety. And let's watch this video. Maybe.
The cell prepares for meiosis just as it does for
Totally
mitosis.
1950s.
But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father.
Mother, father.
Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two.
Sounds sad music.
The sisters are leaving.
This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
Totally 1950s, but I wanted you to see it. So that's one way that you have variety. The second way is how they line up at metaphase. Okay, here we go. So here we can see the homologous pairs, and I don't see any evidence of crossing over in this particular picture, but it's showing you synapsis. And when they do the crossing over, you have exchange of chromatids. And then, now, here we go. The second way you have variety is the law of independent assortment. And this picture is showing you 3 homologous pairs. How many chromosomes total? 6 chromosomes total. And they could line up this first way where all the blues are on the left and all the reds are on the right, or they can line it this way, or this way, or this way. One homologous pair, when it lines up in the middle, doesn't tell any other homologous pair how to line up. And this is why, again, what did we say that you are? As a product of your parents, you are 1 in 70 trillion. Because what you are is you could have crossing over, which could lead to variety. You could have independent assortment that could lead to variety. And the last way is because you don't know what sperm is going to fertilize that egg. So the third way, or independent assortment, boy, I'm really screwing this up. Independent assortment, each parental homologous pair can line up either way during metaphase 1, that's number 2, can line up either way during metaphase 1. And then number 3, fertilization is variation in which sperm will fertilize which egg. Variation in which sperm will fertilize which egg. Okay, so now answer the question. Why are the puppies different than the parents? Not it. Pass or play. Go. She give 3 reasons.
No.
All right. So now you're going to be if chromosomes were licorice, you're going to be comparing and contrasting mitosis with meiosis. So you have mitosis over here on the right and meiosis on the left. There are 2 divisions, they're just shortening it up here in this image in meiosis. Here on the right on mitosis, you can see the chromosomes becoming visible, single file, you separate the sister chromatids, done. Clones of the original parent cell. On meiosis here on the left, you can see crossing over, evidence in the first picture. The second picture, they have lined up this way, but they could have just as easily have lined up all the blues on one side and all the reds on the other side. That's independent assortment. You're seeing anaphase 1, the homologous pairs are getting separated. And now you have 2 technically haploid cells at this point because you've separated the homologous pairs from each other. Then you're going to undergo meiosis 2, and that will leave you with 4 haploid cells. On your notes, I gave you a little chart on number of divisions. What do you need to put in the mitosis box for number of divisions? 1. Good. Could Slate explain the meiosis box? You filled in the mitosis box with number 1, but could Slate explain the meiosis box? Go. Okay, now discuss with your bio buddy, how should you fill in the next boxes? Take a look at that. How should you fill them in? Okay, come back to me. How many daughter cells do you produce in mitosis? How many? 2. How many in meiosis? 4. Good. Fill that in. The results for mitosis, diploid identical cells. Diploid identical cells. And for meiosis, haploid and not identical due to, and what makes them not identical? Okay, name 2 things. Crossing over and independent assortment. The location for mitosis, all tissues for growth and repair. And meiosis, reproductive organs. Reproductive organs. All right, and now we have a slight difference between meiosis in males and females. Okay, now males, when you do meiosis, you are constantly generating thousands and thousands and thousands and thousands and thousands of sperm. Within this class period, tens of thousands of sperm you have all generated, gentlemen in the room. Okay, females, we're in a little bit different situation. Females are the ones who are going to be receiving the sperm, and the offspring is going to develop within that female body for internal fertilization. And for us, how long does that take about? almost 10 months, 9.5 months or so. And so that development has to take place. So when the egg gets fertilized for us, it happens in the oviduct, and then it has to travel down into the uterus, it has to implant, you have to hook up with the placenta. And so what we tend to do is not make 4 sperm like you do, make 4 haploid gametes. What we do is an uneven meiosis 1. And so we don't spread the cytoplasm equally into each cell. We make one big cell and one itty bitty cell called a polar body. And then that polar body may or may not undergo meiosis 2. So this is meiosis 1 and this is meiosis 2. This polar body may or may not undergo that second step, but it's going to make 2 more tiny cells. And over here, what happens is we don't undergo meiosis 2 until an actual sperm goes and starts to penetrate the egg. Then we undergo meiosis 2, and we again concentrate all our cytoplasm in one large ovum and then make another little polar body. So in that way, it has enough nutrients to make the journey for us down, the oviduct and implant in the uterine lining. Okay, so it's a different approach. Whereas the male approach over here, your idea is to inseminate as many as possible and generate as many possible sperm as you can. So just a biological difference. Okay, so on your notes, forming of sperm is called spermatogenesis. Genesis means beginning, so beginning of sperm. And it's where does that take place for humans? In your testes. You didn't want to say it. Testes. And it results in how many haploid sperm? 4.
Why did they put test? Why is it test?
Testes. And then oogenesis, it results in how many eggs?
Testes.
1 egg. And the location is initially in the ovary, but it completes the process in the oviduct. Cytokinesis does not occur until the sperm penetrates the egg. Cytokinesis is done unevenly to keep the nutrients in one egg cell. Unevenly to keep the nutrients in one egg cell. 2 to 3 remaining smaller cells called polar bodies will deteriorate. Not it. Whoever is not it, could you pass or play on differentiating between spermatogenesis and oogenesis, please? Number. 'All right, guys, here we go. Number. Let's go for some hundies. 19 number. Five, 4, 3, 2, 1. Oh.
Wesley.
Unmellow yellow. Okay, here's my approach on these questions. I see the word separate, so immediately I'm thinking of one stage only. What is that? Anaphase. Then I just need to know what's separating. If it's the homologous pairs, right, then it's got to be anaphase 1. If it's the sister, pairs, homologous pairs, parents, divorce first. And then if it's sister chromatids, it's got to be anaphase what? 2. Be better.
That is helpful. Should be better.
Three, 2, 1. Okay, so if the nuclear membrane is rebuilding, it has to be a what? Telophase. And since it says for the first time, it has to be telophase 1. That's how I would approach it. Good job, Carnation Pink. Do these look like quiz questions to you? I agree. They do. They look and smell and taste just like a quiz question. Three, 2, 1. There's only one place where that occurs. This is something you just have to know. It's going to happen in prophase 1 because they're done by the time you get in metaphase 1. All right, so people who chose poorly chose prophase 2. Why can't it happen in prophase 2? No homologous pairs, nothing to cross over with. And then 2 people said metaphase 1. It is done before they line up in the middle. Yes?
Wait, so then where is the law of independent one come in?
That would be in the metaphase.
Okay.
Yeah. Three, 2, 1. Yay! Good job, cotton candy. What's your clue on this one? Yeah. When what else? Yeah, but besides the again. Yeah, and who's breaking down? Nuclear envelopes. You know when the nuclear envelope breaks down, and on top of it, it's doing it again. 3, 2, 1. Okay, so I wouldn't pick prophase 1 because of the word again, but I would pick a prophase. I would not pick a metaphase because by the time metaphase rolls around, the nuclear envelope's gone. I would not pick anaphase 1 because the nuclear envelope is not, it's way gone at that point. Okay, so the nuclear envelope and nucleolus disappear in a prophase. Okay, so I see the word separating, and what comes to mind whenever I see separating is what? Anaphase, and it's homologous pair separating, so it's going to be anaphase 1, but somebody picked prophase 1, and that would be Caribbean green, and whoever you are should have a nice big shame bell.
Yeah, it wasn't.
I promise it wasn't.
It wasn't.
What are you working on?
I'm just
Yeah.
trying
He's
to get a
adding
good
questions to
flavor
our
for it.
notes.
Yeah.
Like,
Sweet.
he is doing a good job. That's mine.
Oh. Oh God.
What directs you to which part of PMAT you're at? Prophase, metaphase, anaphase, telophase, PMAT, what you don't want your dog to do. Those puppies, don't pee on the mat. Okay, so guys, if it says homologous pairs, okay, and the key word here I would have gone with is middle. So if there's middle, I know I'm talking about a metaphase. So I'm either talking about metaphase 1 or I'm talking about metaphase 2. I only have the homologous pairs when I'm in metaphase 1. Okay, because homologous pairs separate metaphase 1, sister chromatids separate metaphase 2. Oh, and mellow yellow. Twice you kept us from Mondays.
Feel ashamed.
What?
Feel ashamed.
Feel ashamed. Three, two. Okay. So the key word there is middle, so I go for metaphase, and it's a single chromosome, so I'm going to go for metaphase 2. Is that, let's see.
Dude, I'm all very.
Sunset Orange, it was a new player, a new failure. All right, so you have charts, graphs, things in your book too to help you review. If you're getting ready for your quiz, do the practice questions at the end. Yes, sir?
Can we go back to those questions? Because I want to see, like, you know how you give us those tips, like middle, separating. Are there
Yeah.
any others for the first?
Well, I can do that during the review session, but we got to move on now. Okay. All right, you're super smart.
Cool.